Great. Well, it's really excellent to be able to come on after Chris's inspirational talk. But actually, both Chris and Nick really highlighted some of the challenges that the industry faces and finding new medicines faces. And I guess I was asked here to give a pharma view of this same set of problems. So I'm a neuroscientist, and I'm not embarrassed about that. And so I'm going to talk mainly from the perspective of neuroscience because that's what I know best. But I think neuroscience highlights many of the problems that other therapeutic areas and challenges for diseases experience. So, and I think the things that have really changed recently in drug discovery and finding new medicines is the bar has been raised, and Nick pointed this out. The bar has been raised in terms of things like safety. The bar has been raised in terms of value for patients, which is obviously really, really important. But the problem for then finding new medicines is you've got to find something substantially different. And usually that means you've got to find a new mechanism. And large pharma companies are really, have been in a very bad place for finding really, truly innovative new science and new medicines as the bar gets raised, because they're large static organisations. There is a relatively low turnover of people, and as we all know in science, particularly, science moves incredibly rapidly, moves more and more rapidly, and so what you really want is a very flexible group of scientists who all have the same ultimate aim and are fairly organised, but they bring lots of different expertise. And so, I think the reason why big pharma companies have started to, in recent years, really start to make a big effort in collaborating externally is exactly because of this problem, is the access and the ability to collaborate with the smartest people on the planet to solve the problems, as opposed to trying to reinvent the wheel internally, which generally doesn't work very well in pharma. And that's why I'm in my job. I work for Johnson & Johnson Innovation, and my sole mission in life is to help J&J collaborate with the outside world and find the best collaborative opportunities to solve the problems that, for my particular role, that neuroscience face in terms of CNS drug discovery. And at the moment, J&J CNS drug discovery is focused on mood disorder, so hard to treat depression, treatment-resistant depression, for example, bipolar depression, and other severe mood disorders associated with other psychiatric conditions, and also neurodegeneration, particularly Alzheimer's disease, and now also more increasingly Parkinson's disease, and potentially other rarer forms of neurodegeneration. So these are non-trivial diseases to solve, but of course all diseases are non-trivial to solve. But the problem for CNS in particular is that the successes have been very, very scarce, and so we also face a challenge of people maintaining faith within drug discovery and pharma industry in actually keep on putting the money in to actually enable us to discover new medicines. And I think we all know, I'm sure almost everybody in the room has a family member, a close friend, or has known a family member or close friends who've been affected by these disorders, myself included, and they're horrible, terrible disorders. And so just from a purely point of view, personal point of view, you want to bring new medicines. So part of what I'm passionate about, and I think most of the neuroscientists within pharma about, is keeping the faith that we can actually discover fundamentally new medicines to solve these horrible diseases. Okay, so having said that, I will now go back on script, on piste. Hopefully this is going to work. No, that must be a bing. Okay, let's try that. Okay, good. So what is the central problem within pharma that one faces? This is the problem we face, is how do you persuade the head of the company to invest upward of $100 million in a phase 3 clinical trial for your disease of choice? And in our case, this will be say, Alzheimer's disease. If that's Alzheimer's disease, you're talking about upward of 500 million. So this is a really big problem, right? So you've got to face somebody up the top of the pharma company who may have limited amounts of science training, maybe a finance training. They're looking and they're saying, I'm going to sign a check for $500 million for a phase 3 clinical trial on Alzheimer's disease, and nothing has worked in the past. Or I can sign a check to run a phase 3 clinical trial in a slightly new version or formulation of the insulin, which will pretty much guarantee to make us some money. So that's the central problem. So how do we solve that? And that's really why we want to collaborate and reach out. I think, again, Chris identified beautifully how the industry is starting to think differently about these problems and how we're trying to bring in the patients and the disease in the human as early as possible to really try and solve this problem. Okay, so many of you will have seen these types of images before in various analyses of lack of success in or difficulty in discovering new drugs. This is from let's try that. Ah, there we go. So this is from 2004. This shows, the success rate of finding of launching a new medicine from the start to the finish. And it sort of averages around 11%. CNS is about 8%. This has actually got slightly worse. And oncology, which was worse than CNS, has actually got a lot better recently. And that's actually because oncology has been able to make that link into patients much sooner. You can analyse tumours now, understand the mechanisms of driving tumogenesis, and you can then sort of specifically address those in individual patients. So that's really important. So this is the one major problem. If you actually another sort of analysis here actually puts the overall success rate from a target discovery right through successful launch of 1.8%. So these are real problems, because if it costs about $1 billion to make a medicine from start to finish, from an idea through to successful launch, and say only 1 in 10 of those medicines is going to actually be successful at launch, you've got to make $10 billion from every single medicine, so to just pay back your pipeline. That's before you start paying, your investors and stuff as well. So that is really a big problem. Of course, it actually looks like it's worse than 1 in 10. So that's why medicines cost so much as well. I mean, R&D, companies have to recoup the investment they put into these. And as the bar gets pushed higher, the expense goes up. So again, we're really desperate to find ways to make it a shorter, more efficient process. And the other major problem is where the risk resides. Where's the biggest risk? So the biggest risk is actually not in the early part of drug discovery. So TITV, that's target identification, target validation, down here. Lead op, that's when you try and make your molecule better. Preclinicals, when you've got a molecule which you think looks like a drug and you're testing it, safety, etc., etc. These are actually relatively cheap. These are only a few £1 million a year or stuff like that. They're not big bucks. When you get up into the clinical arena, then the cost goes up. And the cost gets very big as you get up into phase 2B, particularly, which is your first proof of concept. So that's the big thing you want to get. And the risk is still not discharged at this point, essentially. So you've done all this investment, and the risk is not discharged until you get a successful phase 2B. So basically, you put in getting on for 75% of the investment in drug discovery before you know it's going to work. And so another part of what the industry is trying to do is trying to bring that risk earlier. Can you get much better evidence earlier on, when it's a lot cheaper, that your medicine is going to work? And the other great thing about that is you can try more candidate mechanisms then, so you can actually move through things faster and faster. And again, Alzheimer's disease is a great example. We've had some very high-profile failures, and these proof of concept clinical trials have cost hundreds of millions of dollars, have taken 5-6 years to run. And so you can only do one of those in one big company probably every 10 years, maybe 2 every 10 years, if the company really likes Alzheimer's disease. So it really slows down the rate of actually finding new medicines, usually. So part of what we're really interested in is, can you come up with a better experimental medicine design to discharge risk early? Okay, so essentially how you try and address this challenge is reduce the risk of failure in phase 3. And so the key things that we focus on at the moment is targets. How do you find a better target? So this is getting evidence from the human disease, absolutely critical. Target identification does not come from animal models, cell models. It only comes from the human disease. It makes sense, right? That's what we're trying to solve. Biomarkers. So how do you stratify patients? Again, Chris identified this. How do you make sure you're putting the medicine into the right strata of patients that are most likely to respond? How do you identify, and how do you identify disease mechanism progression? And could that also kind of help you identify early efficacy of a molecule which slows down a biomarker of disease progression, for example? And then clinical trial design is absolutely key. So the industry's got a bit tangled up in trying to bring in the FDA and EMA approved endpoints for clinical trial design early on in experimental medicine studies. And you sort of throw away much more sensitive measures. So we're trying to get back to that. You're trying to treat the early clinical trials as another experiment where you use the most sensitive measure in humans for the mechanism you're particularly trying to test. So more sensitive endpoints, absolutely critical. And I'm happy to say Oxford is great in all these areas in neuroscience, and that's why I spend a lot of time in Oxford at the moment, because we are setting up a number of collaborations with Oxford academics in these areas. Okay, so I'm going to give you a few quick examples. I'm going to run out of time, and just do that to me or something. I'll just stop at that point. And the slides are here and available, and please circulate them to everybody so you can look at them as well. So target identification. So again, I'm now talking about neuroscience. So psychiatry and neurodegeneration has really suffered from identifying new targets. You know, a great example is trying to identify new depression targets with animals doing forced swim tests or other behavioural models of depression, which turn out not to be behavioural models of depression, because mice, we don't know if they get depressed or not. So, but really, one of the big things that's come out recently is, of course, the human disease genetics. And again, Oxford has been one of the leaders in this area. And so now, in terms of Alzheimer's disease, these are the genetic risk factors for Alzheimer's disease. So there's some highly penetrant causative ones. These are penetrant mutations in amyloid precursor protein processing, which produces amyloid plaques, which is one of the hallmarks of Alzheimer's disease. And then there's a bunch of GWAS hits, which are very obviously low risk, but quite common in the population. And in the middle, there are some low to medium risk alleles, which have reasonable frequency in the population. And so you can use this genetic landscape to then to identify collections of genes that form around particular biological pathways, which then give you ways in to start to identify pathways, and you can then find nodes and ways to manipulate those pathways. So we're very interested in doing that. So we're currently working on some targets around the innate immune system for Alzheimer's disease with the UK Drug Discovery Institute in collaborations led with John Davis, Eleanor de Daniel, and with Sally Cowley and Olaf and Sorge as well from the Dunn School of Pathology. So that's really interesting. And so in that collaboration, we have a postdoc. They've got a postdoc in their lab, and he's working on coming up with an assay for one of these targets, which, if successful, will then enable us to start a drug discovery program at the Drug Discovery Institute in Oxford. And the expectation there is then we'll do an external drug discovery project with Oxford, which hopefully will come out at the end with a medicine, which will be shared IP between Oxford and J&J. So that's the idea. And if everything's great, we can test that at the NHS or the clinical research facilities in Oxford. The idea we could do the whole thing, the whole drug discovery thing in Oxford. And I think that's a real power here. Another really interesting area for target discovery is bioinformatics to analyse electronic health records. So this is, I think, an extremely interesting area. So again, how do you find new targets for disease? So one way is to do the genetics. The other way is to ask the question, in the population, if you take, what modify your risk of getting a disease or slow down the progression of that disease? So, for example, Christopher Edwards, Simon Lovestone, Danny, etc. These guys at Oxford here have done this fantastic bit of work looking at this is the clinical practice records database, looking at a modifier for Alzheimer's disease risk. They found that disease-modifying anti-rheumatic drugs reduce one's risk of Alzheimer's disease on average by 40%. And in fact, methotrexate reduces your risk by 50%. I mean, that's an incredibly interesting finding, and actually it fits beautifully with the genetics, which is identifying the immune system, the innate immune system particularly, as a risk factor for Alzheimer's disease. This is another really interesting study which came out of Norway. They found that in the Norwegian health records that people who take salbutamol for asthma, so that's a beta agonist, have a 50% reduced risk for Parkinson's disease. And people who take beta blockers for blood pressure have an increased risk for Parkinson's disease. And they also found independently in a cell-based assay that the beta receptor modifies the production of alpha-synuclein, which is one of the causative pathologies of Parkinson's disease. And this is stunningly interesting stuff. So this we think is a really, really important area going forwards. more sensitive biomarkers. So I'm going to stick to my Alzheimer's disease example here because this is what I know about best. As I mentioned, the clinical trials in Alzheimer's disease are incredibly expensive and prohibitive in discovering new medicines because they're so slow and so expensive. So we're working with, in a consortium under the Dementia Platforms UK with Keir Nowbray and Mark Woolrich at OBAR here in Oxford. It's actually led by an academic in Cambridge, James Rose, a neurologist, also University of Cardiff involved, also some other pharma companies, to identify whether MEG, which is a form of brain imaging, can provide a sensitive biomarker for disease progression in Alzheimer's disease. So essentially you're going to look at three sets of individuals, control people, people with early Alzheimer's disease, and people with actually cognitive deficit, which is a very, very early form of Alzheimer's disease, MCI, and ask the question, over a year, can you see a big progression in these MEG signals as a biomarker in a relatively small number of patients? And if that's the case, then it means you could then run an early experimental medicine trial for efficacy in about 200 or 300 patients over a year, as opposed to 600 or 700 patients over 3 years, which would greatly speed up the rate of drug discovery. And this study just started last month, so we'll get it's a 12-month study, so we'll start to get the readout about this time next year. And this has been funded partly by Dementia Platforms UK. Actually, most of the money came from us, but also actually the universities have been great here because they reduced their scan costs, for example, and really reduced the costs, their internal costs, as much as possible to make this possible. So I think it's been really a real success story. Another interesting thing around experimental medicine is actually stratifying patients more effectively. And again, Chris was talking about this earlier on. And so in psychiatric disorders, I think this has been a real problem. So in psychiatry, there's very rarely hard objective biomarkers for disease. Essentially, the classification of a disease, schizophrenia or depression, is based on the clinician's interaction with the patient, and the patient essentially it's mainly self-report. And so, interestingly, what we did is we actually went back and reanalyzed some data in collaboration with GSK on an anti-IL-6 antibody, which is sirukumab. I keep pressing the wrong button, sorry about that. Sirukumab is an anti-IL-6 antibody that's used peripherally to reduce inflammation. But there's been a hypothesis out there for a number of years that peripheral inflammation can drive central inflammation and also a central depression phenotype. And indeed, we found that, first of all, that the cytokine-based immunotherapies like interferon actually cause a depression phenotype. That's been known for a number of years. And we found that cytokines are elevated in patients with treatment-resistant depression, and the levels of these cytokines, particularly IL-6, correlate with the depression's severity. And then in this reanalysis of the sirukumab data from a clinical trial, which is on rheumatoid arthritis, we found that the depression score, which was routinely measured in these patients, is reduced by sirukumab in patients who had this elevated IL-6. So although this needs to be confirmed in additional studies, this retrospective study indicates, I think, for the first time, that there maybe is a real very strong link between peripheral inflammatory cytokines and depression. And so one can imagine that you could then stratify people who present in the doctor's clinic with depression with a simple blood test and then select the right treatment. Okay, I think I'll stop there because I'm getting stern looks. I said that the slides are available, and particularly at the end of the slide pack, I've just got a couple of slides on what we, from the pharma point of view, consider when we're looking at a collaboration with an academic group or a group that's trying to spin out an opportunity into a small company. And then from your perspective as the scientific originator spinning out the company or trying to interact with pharma, what do you want to get out of it, which is really important to us. So we only really want to work with people